Hi, everyone. I'm Katie Sacra, the Family Education and Engagement Liaison for the Global Foundation for Peroxisomal Disorders. Thank you for joining us for today's webinar, Peroxisomal Disorders 101. We will take time at the end to answer any questions you may have. Please feel free to submit questions via the Q&A section, which is beneath the player window, or through the chat box feature. A quick reminder that this webinar is being recorded. Our presenter today is Dr. Wangler, who many of you may know from GFPD conferences. Dr. Wangler is a pediatrician and medical geneticist interested in childhood metabolic and neurological disease. He is an assistant professor in the Department of Human and Molecular Genetics at Baylor College of Medicine. He is Texas Children's Genetics Clinical Investigator in conjunction with the Jan and Dan Duncan Neurological Research Institute. Dr. Wangler had the honor of doing a pediatric residency at Texas Children's Hospital under the late Dr. Ralph Feigin, whose interest and enthusiasm for genetics has been inspiring for Dr. Wangler. Now the Wangler Lab works according to the general principle that studies of patients with rare forms of human disease or where some of the strongest insights into human health have emerged. The Wangler Lab studies animal models, particularly fly models of peroxisomal disorders, and uses metabolomics to identify new biomarkers and treatment target pathways. Dr. Wangler is passionate about using exome sequencing to identify novel and unique atypical subtypes of peroxisomal disease. Dr. Wangler, thank you for joining us for this webinar, and I'll go ahead and turn it over to you.
Okay, thank you everyone, and thanks so much for having me. And I hope we're gonna have an educational webinar today. So I'm gonna start by going over our objectives, the objectives of our time here today. So we're going to spend a lot of time talking about peroxisomal disorders and diagnosing them, including the traditional, and then we'll go into kind of what's happening now in terms of the diagnostic strategy. And then we're going to cover the types of peroxisomal disorders. So in the past, we've had a lot of individuals who are interested in the Zellweger spectrum disorders, and so there's going to be a lot of comments about those, but we will discuss kind of all the types of peroxisomal disorders. And then we'll go into terminology. We will spend some time talking about severity and forecasting severity. And we'll be covering some of the specialized lipids that are inherent in these conditions. We'll talk about PEX genes, variants, the inheritance, and then we'll discuss some of the research and the symptomatic treatments. So, so to introduce peroxisomal disorders, I'd like to introduce inborn errors of metabolism. So, Archibald Garrod was actually a physician who was the first to identify what we call Mendelian genetic diseases, so single gene disorders in human beings. And when we say single gene, what we mean is that having a mutation just in that gene determines entirely the trait and the risk of the person having disease, as opposed to things like high blood pressure, where we think there's a lot of genetics, but it may be many genes that are adding up to a person having that medical condition. And Dr. Garrod's concept was that there were medical conditions that were called inborn errors of metabolism, where the body lacks a specific enzyme to do body chemistry. The way that enzymes work are that we have these biochemicals in our blood, in our tissues, within our cells, and those we call substrates. And then we have enzymes that facilitate making a product out of that substrate. So what you can imagine is that if an individual has a defect and they lack the enzyme, they actually would have buildup of the substrate, and maybe that would cause toxic issues, and they would lack the ability to make the product. And that is basically our understanding of a lot of the issue that comes with having a peroxisomal disorder. I'll add, though, that there's kind of this complication that in the case of peroxisomes and peroxisomal disorders, the enzymes are within this sort of package. They're within this packet called the peroxisome, which is an organelle. So let me introduce organelles a little bit. Um, but before I do that, I'm just going to mention that we'll just mention the genetics of these inborn errors. So the enzymes that Gerard sort of came up with and that we found subsequently are basically they're proteins, and they're all encoded by our genes. So we have genes for many, many thousands of chemical reactions that occur in our body. And genes can have different versions, which we call alleles. So some alleles could be a mutation that would basically lead to loss of function where the person wouldn't have that enzyme. And we inherit basically one allele or one version of a gene from our mom and one from our dad. And so we're going to cover this in more detail later in the talk. But most inborn errors and the peroxisomal disorders that we're going to be talking about today follow what we call an autosomal recessive inheritance pattern. And so what's happening in a recessive disorder is that you have sort of the dad and the mom, and each individual has 2 copies of every gene is the key here. So if the dad sort of has one working gene and one non-working gene, maybe the allele with the mutation that we mentioned, then, and the mom, let's say by chance, has within that same gene the same situation, that couple now has sort of this risk in which a quarter of the children will be unaffected, and they will not carry either of the mutations from the mother or the father into future generations. A half of the children will be unaffected. They'll be in the same basic genetic scenario as their parents, where they're a carrier for that condition, but they themselves are not affected. But then in 1 in 4 pregnancies, the child would be affected because they actually inherited 2 non-working copies of the gene. And these probabilities, by the way, are with each pregnancy. So it's like flipping a coin each time. You could have a family that has 4 children and all 4 could be affected. You could also have a family with 4 children where, you know, there's sort of 1 who's unaffected, 1 affected, and 2 carriers. So, so in the case of the peroxisomal disorders, we are dealing with those defects, those autosomal recessive genetic mutations, but they're all sort of centered on this organelle that is called the peroxisome. So what are organelles? So organelles are basically where our cells within our body make these unique compartments. And if you think about this, this is very important for the way our body works, because our cells might want to do very special things in very unique places, and it doesn't want to cause damage or other effects to the rest of the cell. So it sort of puts machinery within a compartment, and then it just does that specialized work in that compartment. And what you'll see, what I hope I'll emphasize to you, is that the peroxisome is a very specialized compartment. It has very special role in our body. And basically what this means is that you actually have to target all the machines to get to that place. So if your cell's making in a compartment, it's got to get all the proteins to that compartment. And that compartment will have trafficking where it moves, or it will communicate with other compartments. So this is really pretty complicated in terms of how peroxisomes have a role in our body. And so if we think about what is a peroxisome, it's basically what you can see here is a schematic of our cells. And you can see that there's different kinds of organelles, right? There's these different kinds of compartments. And each of these compartments has its own special role. And it's going to have a unique kind of microenvironment where you put the things that you want to do there only in that place. And the peroxisomes we have here depicted as these blue structures. And what we'll see as we go through the talk is that peroxisomes are, peroxisomal disorders are basically due to different classes of problems within that little compartment within our cell. So it's just such a tiny thing, but it's so important for the working of our body. So let's talk about peroxisomal biogenesis. So what does that mean? So peroxisome, hopefully I've kind of explained what a peroxisome is. It's this unique compartment within our cells. But biogenesis is basically the term saying how the body makes the peroxisomes. So it's sort of the synthesis of peroxisomes. And our genes, our genome, within our genome contain a number of evolutionary conserved proteins. And those proteins then are sort of tasked with making this site within our cells. So one thing I like to think about is sort of like, think about a factory. So you make a specialized factory and it has a very specialized role in a town, for example. And in order to make that factory, you actually have workers that will go and build the factory itself. And that's the peroxisomal biogenesis. And so if you start to think about it, you could realize that you could have a problem in the factory where the workers don't show up. And that could be one type of peroxisomal disorders. But if the workers never showed up to make the factory, then there's other issues, because you might be perfectly okay at making the factory workers, but they don't know where to go because the factory was never built. So, so the idea is that biogenesis is basically the generation of this organelle from scratch. And without this, the enzymes really don't know where to go. They don't have a target. So this kind of shows a picture of what I'm kind of trying to explain to you. So the bubbles are peroxisomes, but what's interesting here is that what you can see is out here on the right, this is the fully formed peroxisome. So this is what we want to happen, is the final step. And this is the machinery that's going to get us there. So you can see that there's these PEX genes that encode these PEX proteins. And then within these proteins basically create a series of steps where they turn this bubble that starts out as sort of just a bubble of lipid into a peroxisome. And there's key steps within this process. And then a step that we'll spend a little bit of time on is that the body makes the enzymes. The enzymes are the things that will do the work. And those actually have to be made and targeted into the peroxisome in order for everything to function. So if we take a closer look at a similar process or at this process, sort of what's shown on the left is kind of what I just showed you, the overall process of making these peroxisomes. But this shows you a little bit more of the complexity, how intricate it is to actually get the enzymes in. So the red hexagons depict our peroxisomal enzymes. And what you can see is that there's specific PEX proteins. They're going to go grab those enzymes, drag them to their target, and then they're going to sort of push them inside the peroxisomal matrix. So, and there's different actual targeting systems that we call these PTS systems that allow those enzymes to get there. So, you know, it's not crucial for you to know all the details of this process, but I just want you to kind of understand how there's these intricate machines that are basically building this site from scratch and moving the enzymes into that site. So once you make a peroxisome, what's so great about a peroxisome? Why did you need to go to all this trouble, have all these PEX genes just to make this site? So this is going to try to give you an overview of what's happening in the factory. You know, why do we need this in our bodies so much? It's difficult to, you know, kind of, it's difficult to boil it down so much, but one thing you could think about is things that need to be broken down. So you could have like waste products that need to be taken away, and then substrates that need to be synthesized, meaning you need to make these things for your body to function. And so basically the the key that we're going to talk about are the very long chain fatty acids. Those are lipids that we really need peroxisomes to break down. And then bile acids and plasmalogens, those are key lipids that you need the peroxisome to make those. At the bottom kind of shows a little bit more detail where the very long chain fatty acids and things like phytanic acid actually go through the same system. The same enzymes do the same work for these different types of fats. But the real take home is that there are common clinical tests that we can do in plasma or in red blood cells that will tell us if these things are working. So we can measure very long chain fatty acids. We can measure the phytanic acid. We can measure the bile acids and the plasmalogens. And that's really going to tell us a lot about whether the peroxisome is working. And if a part of it isn't working, it's going to give us some specific information about which part is not working. So I'm going to spend a little bit of time on very long chain fatty acids because those are invariably going to come up in sort of any diagnostic process. So these are, our body has all kinds of fatty acids, and fats are just sort of carbon chains. And very long chain fatty acids are just, all it means is that there's more than 22 carbons. So it's longer than 22 carbons. So carbon 24, carbon 26, those are examples of very long chain fatty acids. Within our bodies, that subtype of fat is much less abundant than, say, carbon 16 or carbon 18 that we might get from like palm oil. And they're synthesized in our body through special pathways, but they're very important for instance, the white matter of our brain. So very long chain fatty acids are going to come up over and over. And so this is kind of an example of a patient with a Zellweger spectrum disorder. And you can see that these are the types of results that you could get from a very, very long chain fatty acid analysis. So basically what's being shown here is here's the patient and the results that were measured. And then it usually comes, these reports usually come with sort of control and Zellweger mean plus, a standard deviation. So the key thing that I always look at that's very important is these ratios, okay? So the C24 to C22, the C26 to C22, those are really strong markers of peroxisomal, of a peroxisomal problem. And the reason is because the C26, as I sort of mentioned, you need the peroxisome to break that down, but the C22 should be able to break down on its own through another pathway. So if a patient has a problem in their peroxisomes, that C26 to C22 ratio is going to go very high. And you can kind of see that here with that in a normal individual, that ratio is kind of 0.01. So it's a very tiny amount of C26 compared to C22. But in individuals with these disorders, you see a pretty big elevation. So the key here, again, is not just to give you some taste of the detail so that you kind of understand where these determinations are coming from. Then, the other thing I would mention is that if you sort of add the very long chain fatty acid testing to the fact that we can test these other peroxisomal functions. So mentioning that we have plasmalogen lipids, and these are unique lipids within our myelin sheaths in our brain and other membranes, and we can actually measure these directly. Bile acids, important for fat-soluble vitamins, and then the breakdown of branch fatty acids, and then protecting our body from oxidative damage. So we can actually measure all that, and that gives us a very clear picture of kind of what's going on with the peroxisome. So if we sort of go back to this and just think more globally now, is that if we have inborn errors that could result from peroxisomes, you can actually think about that there might be several types, right? So there's all these different specialized roles of the peroxisome. So you could be lacking an enzyme in one of those, or you could have a PEX gene mutation. And the PEX gene mutation would mean you never made the peroxisome in the first place, and all those enzymes sort of lacked their targets and they didn't go where they were supposed to go. And that's exactly what we see. So that this is kind of showing different classes of peroxisomal disorders above. And so you can break them down into sort of two major classifications, two major categories. You'd have biogenesis. Let's see. Is there a is there something in the chat that you all can't see the slides, or everything's okay?
Everything's okay.
Okay, sorry. So, you'd have sort of the class of biogenesis disorders. That's the PEX scenario. You can't make the peroxisome properly, so the enzymes don't know where to go. And then the single enzyme defects are like just that. They're a single enzyme that's missing. And so the point I want to show you here is that if you kind of look over in this side at the single enzyme defects, these patients have problems in one pathway, but they're normal in kind of all the other pathways, right? Whereas when we look at biogenesis disorders, there's sort of a global change in all the pathways. And that's kind of what you would expect. So that's kind of summarized here, where we say there's single enzyme or protein deficiencies, and that's basically affecting a part of the peroxisome. So there's, only a part of the peroxisome not performing the job that it's supposed to do. All the other things are there. So when we do tests, we see, well, this part looks okay, this part looks okay, but this part is defective. And then in the biogenesis scenario, we actually see effects that can happen in the entire assembly of the peroxisome. So the peroxisome itself is just not being made properly, it's not working, or the targeting is not working. So so that kind of explains the two major classes that we talk about. But another key issue that we'll cover in some detail is what we call the spectrum. So not only can you have these different types of peroxisomal disorders, but then within the types, there's going to be a spectrum of what exactly happens. How does it affect the patient? How does it affect the individual to have that issue? And that could depend on the specific gene defect and even the specific change in the gene. So we'll cover that. So here's an example of a spectrum. And what I want to emphasize here is that if you look in medical textbooks, you'll kind of see, especially older textbooks, you'll kind of see a classification that breaks down like this. And you may see, sort of photos that are like this, where there's sort of disease number 1, classic Zellweger syndrome. By the way, what's being shown here is kind of a marking of some of the abnormalities of the bone structure, enlargement of the liver and spleen. But you can see that this child kind of has some facial features that are abnormal, and this particular patient was sort of noted to have low muscle tone and seizures right at birth. Then, sort of disease number 2 was called neonatal adrenoleukodystrophy, and that was because sort of infants were having changes in the brain MRI that looked similar to people as adrenoleukodystrophy. And then this individual was diagnosed with infantile refsum. And what we now understand is that all these individuals are all part of a clinical spectrum of disease. And we call that spectrum peroxisomal biogenesis disorder, Zellweger spectrum disorder. So we're getting rid of the classic Zellweger, neonatal adrenoleukodystrophy, infantile refsum. and we're just going with PBD ZSD. And then we see severity spectrum within PBD ZSD, where, you know, sort of today, if we were seeing this child, we would probably say this is severe ZSD. This child would be moderate or intermediate, and then this child would be mild. And we can also notice that that severity spectrum tracks along with the biochemical phenotype, meaning that if the very long chain fatty acids are really elevated, that would actually predict a somewhat worse scenario than if the very long chain fatty acids are normal. And then we'll go into the different types of alleles. Remember, the allele was the version of the gene, the specific mutation. And so, you know, a patient who has a really severe mutation, that may differ from a patient that has a mild, we call it hypomorphic mutation. So I just wanted to, this was actually from our own research, and the reason I wanted to show it is, so this was a study where we actually looked at a number of patients who the GFPD helped us conduct this study, and the majority of the individuals in this cohort had PEX1 mutations and were in either the intermediate or mild range. And what's being shown in the columns is this is each individual with a peroxisomal disorder, and then across the rows are hundreds and hundreds of biochemicals, amino acids, peptides, carbohydrates, energy metabolites, lipids, nucleotides, cofactors. And so what you can see is that there's a distinct pattern. And the point I'd like to make is that the global change in a peroxisome has a pretty dramatic impact on, you know, hundreds of biochemicals in our body. So, I mean, this we were able to focus really on more a number that was, you know, closer to 20. But you can see that there's a lot of effects that can come about. Some are consistent across the groups, and some can be specific for specific individuals. So you can see differences as well between specific individuals. So so let's just kind of review, because there's a lot of material. You know, by the way, I didn't start out by kind of warning you that you know, I sort of took my approach to this lecture was I took you know, a lecture that we give to trainees in, you know, medical genetics, and I sort of watered it down only enough that you would be able to attend this webinar, and I'll explain my you know, my rationale for kind of really giving you all the detail later in the talk. But, you know, so I just want to emphasize that I know it's a lot of detail for you to take in. But this is a great review slide, kind of that will kind of bring it together. So we sort of can divide the terminology, and we should really just talk about, you know, this should be the terminology that we adapt. And we've obviously argued from the GFPD and the scientific advisors that this is the new terminology, is that we sort of have two classes of disorders, the peroxisomal biogenesis disorders, the PBD, and there's two major classes of those actually, and we'll go a little bit into that, the Zellweger spectrum disorder, and then the rhizomelic chondrodysplasia punctata, we'll just call it RCDP from now on. And then there's single enzyme deficiency disorders. And today we are going to mention X-linked adrenoleukodystrophy, REFSUM, and a few others. And the reason that this is important is because, you know, historically, it sort of is the same group of patients that were historically termed Zellweger, neonatal adrenoleukodystrophy, infantile REFSUM, Heimler syndrome. So you may read these older terms, and if you do read these older terms, you know, I'd encourage you to kind of pull out this graphic, and you know, it sort of helps you calibrate what's being talked about. the way it sort of comes about is that these were all recognized before we understood the genetics. Now that we understand the genetics, we can kind of see a pattern and a spectrum much clearer. So, so let's talk about, we're going to spend a lot more time on Zellweger spectrum disorders, but I want to cover RCDP. So peroxisomal biogenesis disorders, rhizomelic chondrodysplasia punctata, RCDP. And this kind of emphasizes that not all biogenesis disorders are the same. So I showed you a series of 3 patients on the Zellweger spectrum disorders. And what we had there were different classes of brain issues, liver problems. There were some kidney issues. But RCDP is kind of a wholly different looking disorder. So patients with RCDP have dramatic foreshortening of the limbs, particularly the proximal part of their sort of long bones. And you can see that the individuals are short in stature. They can have cataracts of the eye and a lot of brain issues. So why are there these different kinds of biogenesis disorders, and why are they so different? Why aren't they kind of the same? And the key is that if we think again, if we go back to our schematic, there were different kinds of targeting signals. So there's ways for the PTS1, so there are ways for a lot of the enzymes to get into the peroxisome. And that was sort of through this PEX5 pathway. But the issue in RCDP is usually PEX7. So PEX7 is kind of the key to that mystery. If you're missing PEX7, you're missing the PTS2 system, but everything else actually runs just fine. So RCDP is unique in that those patients have normal, very long chain fatty acids. It's just really the plasmalogens that depend on the PTS2. There's a couple other pathways as well, but the key is that this is still a biogenesis disorder because it's still a problem with making the peroxisome complete, but it's very different from Zellweger spectrum. So the take home here is that because of the different sort of traffic that can come about, we can have even different classes of biogenesis disorders. So single enzyme disorders that you may hear about, regardless of whether you're dealing more with biogenesis disorder. One is X-linked adrenoleukodystrophy. And we're going to talk about this in some detail. This is the most common peroxisomal disorder. And it's a pretty specific defect in that very long chain fatty acid transport. So patients with XALD, they can't get the very long chain fatty acids into their peroxisomes, and that's where the problem occurs. D bifunctional protein deficiency is something that we've seen a few times, and it's a very severe disorder of very long chain fatty acid metabolism. There's bile acid metabolism issues in that disorder. And then another example of a single enzyme disorder would be Recessive disease. So these are adults with retinal and hearing issues that would actually come about over time and is due to the buildup of phytanic acid. So that, that's a classic kind of single enzyme disorder. Remember, I kind of mentioned that you've got sort of, you could have a change that affects the whole peroxisome, or you could have a change that just affects that one tiny thing. And Recessive disease would be an example of that one tiny thing. It's just that phytanic acid can't be metabolized, and those patients build up phytanic acid. So I want to mention X-linked adrenoleukodystrophy in a little bit of detail because we're going to talk a little bit about the newborn screening and how it affects the Zellweger spectrum and other peroxisomal disorders. So XALD is, the most common peroxisomal disorder. It's a single enzyme disorder. As opposed to the other conditions I mentioned that were recessive inheritance, XALD has a specific X-linked inheritance. So typically A boy is most affected by XALD. And the issue for XALD is that it can come in three major forms. So the childhood cerebral form is usually a boy who, between 4 and 8, he might start out with a diagnosis of ADHD that might come from the school, but over time, that boy can undergo progressive brain deterioration and early death without treatment. And the major treatment is bone marrow transplantation, which actually can halt the brain progression if it's done early enough. But often these patients, it's sort of a race to diagnose them and then think about doing that treatment before too much progression. So you can maybe appreciate how that's a real time-sensitive disorder. And that was a lot of the argument for doing newborn screening. Like if we could know about these boys, from birth, we could sort of prepare ourselves for what might happen. One thing that's interesting about this disorder is most patients don't have the cerebral form. The majority actually have what we call adrenomyeloneuropathy, and that's more adult onset in the late 20s with spinal cord dysfunction. And then there's some people that have an Addison only, and that's basically just a hormonal disorder. When you look carefully, there's some blurring of these lines, but that's the three major types of patients that we can see. And again, the gene is actually on the X chromosome, so it's typically boys inheriting a mutation from their mom who's a carrier. This kind of shows that there's, I know you all like, it's not a question of reading brain MRIs, but this just shows how dramatic you can have these areas that really light up for within the brain of these boys. And that indicates a lot of sort of ongoing brain inflammation and is why they need early treatment. So something you all should know about is that XALD, based on the fact that we really want to get to those boys early, newborn screening is really rolling out. This shows kind of in the various states of the US, the situation in terms of newborn screening, with some states who are currently testing. Within Texas, we are testing as of August of last year. Some other states are doing pilots. Some states are starting to test in 2021. And then there's another group of states that are kind of looking, I think, looking at the evidence and evaluating. But this has a big impact, as we'll see, not only on XALD diagnosis, because the method of screening is actually looking for metabolites of the very long chain fatty acids. So a lot of the XALD newborn screening is actually going to be predicted to pick up Zellweger spectrum, D bifunctional protein deficiency, and a lot of other peroxisomal disorders. So that's where, thinking about a schematic like this is kind of interesting. So, let's say that you think about two babies. So the baby on the left, let's make the babies sort of equivalent, like they sort of both have a similar mutations, they have similar presentation, they have low muscle tone, seizures, breathing problems, hearing and vision difficulties. They might have craniofacial differences, low set ears, a large fontanelle. And in the baby on the left, let's say that they're born in a state with no newborn screening available, which also would be the status, for almost all the states before 2018, 2017, 2018. And in that baby, you might you could imagine a diagnostic kind of odyssey where, they would go to a doctor, a number of blood tests might be done, imaging, confusing paperwork, I'm sure, and a lot of medical bills before you might get to a very long chain fatty acid test. And in that situation, It is important to emphasize, though, that elevated very long chain fatty acids would suggest, would basically be diagnostic for a peroxisomal disorder, because peroxisomes are responsible for metabolizing very long chain fatty acids. If you have that elevation and it's like a true elevation, you may not be able to tell early on if it's X-linked adrenoleukodystrophy, D bifunctional protein deficiency, ACOX-1, or a Zellweger spectrum. But you certainly know that you're dealing with a biochemical diagnosis of a peroxisomal disorder. And that's where you might turn to additional biochemical diagnostics, red blood cell plasmalogens, other studies, and then you would get targeted gene testing of the PEX genes to get your genetic diagnosis. But now let's think about baby number 2, okay? So in baby number 2's situation, they're born in a state that's currently doing XALD newborn screening. On the second day of life, and maybe again at a week of life, they're going to get this screen and it's going to turn positive for XALD. So there's a lot of issues for that path, actually. First of all, the parents, regardless of what type of peroxisomal disorder they ultimately are diagnosed with, they might initially hear about XALD and then be given all this information about XALD. A problem that we're concerned about with that program is we don't want parents to sort of be told, especially let's say it's a girl, and they're sort of told, let's just sort of keep an eye on that she might be a carrier for XALD. We don't want a patient like that to turn out to have a Zellweger spectrum disorder that we could have done something about. Likewise, we don't want, a family of a boy to be told, well, we really don't have much to worry about before 4 years. So the point being that when we have a positive screen, all we really know is that we have high, very long chain fatty acids, and that could mean many peroxisomal disorders. So I'll say that in Texas right now and in many states, what's happening is that with that positive screen, there's DNA sequencing being done for the ABCD1 gene. So we're able to, from the state lab, kind of tell, whether you have a clear, this is a case of XALD, or this is a patient that the screen was positive, the very long chain fatty acids are elevated. We have to do more workup to figure out why. But in either case, just want to emphasize that positive biochemical test, does suggest that the peroxisome is where the issue lies. What you can see is just there's different pathways to getting to that initial result. And then in the next slides, let's talk about where you go from there, from getting that initial result. So this was in our publication that was a big team publication involving the GFPD on the PBD ZSD diagnostic strategy. And so this kind of talks about that we would start with identifying the clinical features. Maybe we would have that elevated very long chain fatty acid draw from newborn screening. And the evaluations that you would go about traditionally would be, I would check the very long chains in the blood. I'd look at some of the other, remember we mentioned there's other fats that the peroxisome is metabolizing. We talked about bile acids and plasmalogens. So we'd measure all those, again, trying to fill out that table of where are we in the peroxisomal disorder spectrum. And then, particularly in the past, we were actually doing a lot of confirmation in skin. So we were doing skin biopsies on the patients and then growing those cells and testing how those behave in terms of those biochemical assays. And it was only after those kinds of results that we were going to DNA sequencing. Now, I would argue that, that is sort of the traditional path where you go from, I see the clinical features, I look for the very long chain fatty acids, I try to characterize how the skin cells behave, and then I sort of narrow it down and do some DNA testing at the end. But right now, we're in a different era where a lot of this is happening either through the newborn screening. It may happen through carrier screening, where parents are finding out they have a specific PEX mutation as they go in for prenatal screening. Babies who maybe present with seizures or low muscle tone may go straight to next generation sequencing. They may get exome sequencing within the first few weeks of life. And then you'll know about the PEX mutations in those babies before you even know whether the very long chain fatty acids are elevated. So the thing I want to kind of emphasize here is that current changing diagnostic strategy is being driven a lot by sort of the disruptive technology. It's not necessarily that we wanted to build it that way, but it's just as the technology's improved, we've been able to detect these things in newborns much faster. We can find the DNA changes much quicker. And so this disruptive technology kind of puts us in this situation where we have all this information. But then what happens is that, then the families are often left with sort of having to find, how do I find out more about this? What does this mean? So that's really the challenge of this future scenario, is we get the information faster than we know what to do with it. And we have to kind of play catch up and figure out what we're dealing with. So this is kind of the final example that I would sort of give, is that, you could think about diagnosis in the textbooks. If you read current textbooks, the way that they would describe how a peroxisomal disorder would be diagnosed, they'd say a pediatrician may consult a medical geneticist or a neurologist, and then that specialist might say, hey, I think this might be a Zellweger spectrum disorder. They would draw very long chain fatty acids. They would see that they're elevated. Then they do additional tests like the plasmalogens, additional peroxisomal studies. They may do a skin biopsy. And then finally, at the end, they may do a DNA test. Whereas today, a lot of patients are sort of getting whole exome sequencing right after birth, or a newborn screen is showing an abnormality in very long chain fatty acids, but we don't know where it's coming from. We don't know where in the genome is the issue. And then it's at that point that a geneticist is sort of consulted and asked to determine if maybe those DNA changes cause disease. So we're going to talk a little bit about that new paradigm and some of the confusing issues that happen going down that path. So one thing I wanted to try to clear up a little bit is some of the genetic terminology. So particularly if you get into the situation of having a DNA sequencing report done, either maybe this is after biochemical diagnosis, maybe this is happening right away, and you're sort of doing the biochemistry now. But basically, the gene under discussion, that's sort of the hereditary unit. So in these examples, we were talking about the PEX genes. So each PEX gene is going to have its own unit within our genome. And you can have different kinds of what we call variants. So that's a sequence alteration. And it basically is defined as just it's altered compared to the reference genome. The reference genome is something that is an agreed upon reference. It doesn't represent any single individual. In fact, it was sort of collated from about 30 or 40 individuals, but it's considered the reference genome, and then variation is considered alteration from the reference genome. And so you may have scenarios where there's test reports for PEX genes that where the lab says there is a pathogenic or a likely pathogenic variant in PEX1 or PEX6 or PEX16. And what that means is that there's evidence that that variant can cause disease. Remember though, one pitfall is that these are recessive disorders. So you're going to need 2 pathogenic variants in an individual to have a peroxisomal disorder. Now, a benign or likely benign variant, that's evidence that there's evidence out there that that variant doesn't do anything. It's not causing disease. A lot of times the benign or likely benigns don't even show up on the reports. The problem is these variants of uncertain significance. So that's basically where the evidence is not available. And that's difficult for families. Basically what's happening there is we're seeing sequence variation, and we don't have information on it. Perhaps we've never seen it in a patient before, or we've seen it in patients before, but we've never seen it in a scenario that might cause a recessive disorder before. So we don't know whether it's causing anything or is incidental. And a lot of this boils down to the problem of sort of finding incidental findings from these types of studies. So we're going to go back a little bit to that genetic terminology and those diagnostic pitfalls at the end. I'm going to spend, about 5 more minutes covering sort of some of the treatment discussions, and then we'll go back to, we'll just sort of make it practical, and then we'll go to questions. So, so are there treatments? So here I'm going to focus a little bit more on the Zellweger spectrum, but this is just to emphasize that our group and also another group in Europe that's also part of the GFPD sort of worked on these treatment guidelines. So there are published treatment guidelines for Zellweger spectrum disorders. And basically within these papers, we actually have tables with various schedules of different evaluations. We address issues like liver, brain, seizures, hypotonia, feeding, the GI tract, renal. So we sort of address how should we care for these individuals, and what should be the algorithm for treatment. And so I think these are important kind of documents to have on hand, and we'll get to that. We'll talk a little bit more about it in the end. But I think the other thing to say is that, if you read through these documents, a lot of what is listed there would be considered kind of symptomatic care. What you're doing is you're looking at the patient, you're trying to do what you can to improve their life, to address what medical issues could occur, what do we know might occur. But you are kind of, just sort of dealing with each manifestation in and of itself. You're not actually addressing the sort of the peroxisomal problem ultimately. And since that in these conditions, that's the issue, that's the defect, that's what we would love to be addressing. So, I thought I would just mention that a couple months ago, we had a meeting from the Scientific Advisory Board. And, this was just an example of some of the research topics, some of the research that actually some the GFPD is supporting, some is supported by external and public grants. Gene therapy is currently an area of great interest for the group. This is where we would be able to replace a functioning copy of a PEX gene within cells. And there's a lot of issues with this type of approach. We have to package the gene properly. That's not trivial. The gene then has to be delivered to the cells, somehow has to be delivered into the genome of each cell. How many cells do we need? What cells should we choose? What tissues do we need to address? These are all questions that are being looked at, to one degree or another, using the help of different research mouse models, as well as some other methods. Another area is small molecule screens. So if we have, initiatives supported by the GFPD, like at NCATS, at NIH, where basically small molecules, FDA-approved drugs, other drugs that maybe could be approved in the future, are looked at for how can those affect cells from patients with known PEX mutations, are there small molecules out there that could actually help the function of the peroxisomal machinery, even despite the mutation that the patient has? There's some compounds that have undergone various levels of trials in the group. And then, like in our own group, I showed you some of the metabolomic data where we're trying to get a handle on some of the other analytes and body chemicals that are going wrong in some of the individuals, and what role might that play? Could we use that as a marker in some of these treatment trials? So I think just to mention that the GFPD is working very hard to support a wide range of research to help patients with peroxisomal disorders. Okay, so, but I wanted to sort of end with what is some practical advice? So, particularly for newly diagnosed families, what would be some practical advice? Because there's a lot of detail in this talk. There's also a lot of, discussion of research that, who knows how far away the help is from this. So what can a family who's going through this right now, what can you do? This is maybe you could argue, why do I put this first? But I think it's really crucial to obtain your DNA results. Now, by that, I mean, obtain your report. If you've had DNA sequencing, I think you need to get your hands on the report, because that sort of helps you know what you're dealing with. I think it's unfortunate that sometimes I'll see that families are kind of kept in the dark. They'll get the DNA results and then they're sort of told things about what it means, and then not really given much to do. And so I think I would encourage you to obtain your results. If maybe I've understood that some individuals are actually in the part of the diagnostic process where they haven't done DNA testing, and that COVID has really affected that. And that's, that obviously that's something to balance. I don't think it's an emergency to get DNA results. If there's some things that could be done in the short term, just sort of clinically and biochemically. But I think, in the long run, I would encourage each family to, have that report, know what mutations are on it, know what the cause of the, know what the cause of the sort of at the genome level of the disorder is. I also think the treatment guidelines that are published, those should basically be shared with your pediatrician or your doctor, depending on age. I think those are useful. They don't answer everything, but you should sort of share them and make sure that we're on track with what's recommended. So one thing that came up that I wanted to address was severity. So I've had a number of scenarios where we get, say, a DNA report and we have some mutations, and we have maybe a young individual, maybe even a newborn. And if I was a parent, what I want to know is what does this mean? I mean, how severe do we expect this to be? And I have to say that it's easy to kind of fail to really answer this question. Part of it is that it's just very difficult to predict a lot of things. Severity is also something that we really have to define. These disorders have an effect on so many different systems, and we can see some patients that have just terrible problems with, say, liver issues, and other patients that have real, severe seizure issues. And so you have to sort of define the severity of what are we looking at. So the genotypes are also, what I would say is they are unfair. Some families are going to get rich information from that. When they obtain that DNA report, they're going to get rich data from that, because they're going to be able to maybe find reports of other patients who have exactly that same genetic situation, and they'll get knowledge of what happened with those patients at different ages. Unfortunately, other families, they may have mutations that we just haven't seen before. And it'll sort of just be like, you have this information, but we haven't seen it before, so we don't have a lot to say. So the genotypes, what is known about those mutations, becomes crucial for whether we can give you any help in predicting the severity. And then, it's sort of obvious, but there's great variability between individuals. I mean, you saw even in our data for the metabolites, there are individuals with wildly abnormal, sort of specific amino acids or other metabolites. We don't know how consistent that is over time, but it just emphasized to us how much there's individual variability. And then the final thing, so the reason I didn't go easy on you today is, I'm not saying that it should be this way, but I think that quickly as you go through this, you become the expert in these disorders. You become sort of the expert. The doctors and nurses, so we're medical professionals. I'm obviously very invested in these disorders, but even for me, your child, the specifics of sort of where your child lies in this condition, or where, if you're an adult, sort of your own story and where you land in this. And, I'm not going to know those details. So part of the reason to obtain your DNA results, part of the reason I didn't go easy on the details with you today was because, you really are the expert. And I'm not arguing that that's how we should do it, but I think that is how it plays out. So with that, I can sort of end the formal part of the talk, and I can go to questions.
Yes, thank you so much. We actually already have a question from one of our parents who is asking if there's any studies involving diet changes like cutting dairy or beef out on Heimler's, which, as you shared, is biogenesis disorder. So have you seen any diet changes with that diet change? And obviously they're talking about the low phytanic acid diet. So if you could talk to that, that would be great.
So, so the phytanic acid diet is definitely effective in the Recessive disease, where the patient is lacking the phytanic acid breakdown capability. And sort of by logic, we would think that, there's a possible benefit in Zellweger spectrum, because a similar phytanic acid defect exists. It's just that in Zellweger spectrum, there's a lot of other defects that also exist. The evidence hasn't shown that, in that there's not evidence for a big effect on a low phytanic acid diet. I think that many families have done a trial of it. In the treatment guidelines, what I'll say is that I think what we commented is that there's not medical evidence to support doing it, so it's not a recommendation. But there's some rationale for thinking that it's possible that it could help maybe, specific things like hearing loss. And so for some families, it might be an option that they might want to try, particularly if, say, hearing is something that's a major issue that family really wants to try to preserve. So yeah, not a straight answer, but that's kind of how we worded it in the guidelines.
This is Katie Sacker. I also wanted to ask if you could just help clarify, because I know myself from personal experience with my own son who has a peroxisomal biogenesis disorder, that sometimes there's a mix-up with medical professionals when they see that Refsums. They don't know the difference between the old terminology that we talked about, the infantile Refsums disease, versus the adult Refsums disease. And I've had more than one medical professional walk in an exam room and tell me that I need to do this diet and they don't realize that they're confusing those two.
Right. I mean, the terminology confusion is is just, that's why it's a real issue. You know, these were sort of names that were given to clinical entities, clinical diagnoses before the genetics was known. And so, you know, there were some maybe surface similarities between a patient with Recessive disease and a child with sort of a hearing loss and retinal disorder. And so the name infantile Recessive was given. But, you know, I think, yeah, I would try to, I guess I would try to share the treatment guidelines, and that those do have a good discussion of that. The other place is sort of the gene reviews, I'd say. I don't know if maybe we could share that, but you know, I think Nancy had that gene reviews article, and that kind of gives a nice overview of the nomenclature. And actually the slide that we have in here that you guys generated is also something I think, you know, families should have, because it gives that nice historical, you know, bar.
Thank you. Also, I know that we have had some families that have very openly shared that, you know, test results specific to that level is sometimes elevated, and that's when there's a time also. So usually your doctors will be monitoring it pretty closely anyways. So if it does come up elevated, then there would definitely be a discussion about that. I also wanted to share, because you've referenced the treatment guidelines so often, I want to make sure that everybody in our audience knows that if you go to our website, you will be able to go under the area of scholarly articles, and the treatment guidelines are accessible at any time on the GFPD website.
Actually, I see, there's a couple things in the chat. Should I go to those, or?
You can jump in anytime you want.
So one thing,
coming in two different ways.
Okay.
I'm sorry, but if you see one, go for it.
Oh, there's chat and there's Q&A. Wow, hold on.
Go ahead.
Well, let me do chat first, then I'll go to Q&A. So I want to go back. Okay, one thing I didn't explain here is that there's this ABCD1. I didn't even tell you what that was. So that is the gene for X-linked ALD. So just like PEX1 would be a gene for Zellweger spectrum disorder, the ABCD1 gene is the gene for X-linked ALD. So sorry about that. And then there was a question, actually, we could have, when we went into genetic terms, maybe we could have talked about zygosity. Let me go back to this all the way in the beginning, where we were talking about inheritance. So zygosity refers to whether we sort of have two copies of each gene, one that we got from mom, one that we got from dad. And so when we say something is homozygous, it means that the individual has the same copy on both strands. So they actually got the same copy from mom as from dad. And we usually are talking about a specific site in the genome when we use that phrase, homozygous. Whereas heterozygous would mean one copy is different from the other copy. So in the case of like the dad here, he's heterozygous for a mutation. Now, what could come up is, let's say that the dad and the mom both shared a common mutation that they inherited from distant ancestry. So an example would be like the PEX1 G843D mutation. So in that scenario, the child would actually be homozygous for the mutation because they have the same mutation on both strands. So they're homozygous. So it's basically the homo meaning same, zygosity having to do with having the two copies, one on each chromosome, is sort of how that plays out. So what you see is that in populations like in Europe, where there's maybe a lot more shared ancestry, an individual with a Zellweger spectrum may be more likely to be homozygous for the same mutation. Whereas in the States, there's often more admixture, and those individuals may be like, we'd call it compound heterozygous. They have two mutations, but it's two different mutations. I hope that helped. So now I can see the Q&A.
We have a question
Oh, yeah.
about, I may not hear about these, but it seems like there are a few studies in older children with PBDs. I would think it
Yes.
would be good to see if there is some commonality and why some of our children live into their 30s, 40s, et cetera. Is there studies in older children, or is there some reason why there's not?
So there is research on older children with PBD. I mean, I agree with you in that it's a little bit harder to find. So, for instance, often this comes buried in some of the series that have described PEX mutations. So I can send Katie a couple papers. There's one on PEX1 that comes to mind, where they discussed individuals in their 40s and 50s. In the study I mentioned that we got to do with the GFPD, we had a wide age range, and differences in age was one of the conclusions of our study that actually we can't explain, but it seemed like there's some biochemical normalization that happens in adults and older children that we don't see in the younger children. So I think I mean, the main answer is that it's a great point. I mean, we need to be looking across the entire severity spectrum. I think that one of the hopes of newborn screening is that, we really should just collect all the individuals that we pick up at birth, and we should follow them their whole life. I mean, we really need that level of information.
I agree. Thank you. We also have a question about the severity. In discussing severity compared to other children with the same mutation, who or where could we find the comparable of the progressions? Would this come from our genetics department, doctors? This person's own child is a PEX12 mutation,
Yeah.
and they've not heard of many others with this mutation.
Okay, that's a great question. So, I mean, yes, I think practically I would ask the geneticist. In my own work, what I do is there's a database called ClinVar, which is actually a NIH-sponsored database that's freely available. And I look up all the mutations in that database that has kind of links to, if there is evidence out there, that's a good jumping off point for me. Nancy Braverman also has a big catalog of mutations. She's a great person just to email the mutations, honestly, and ask if she's seen them before. I've had situations where Ann Moser, who's kind of retired, but there's cells for certain patients that have specific mutations that we're interested in, and Nancy kind of has access to that information. So that's one of the ways. But yeah, practically, I would get a hold of the report and send it to one of us. You could send it to me, send it to Nancy, ask if we've seen the mutations before, and then. But I would just mention again, unfortunately it's just not fair. So there's some mutations where we'll come back to you with a lot of information, and other mutations we just haven't seen before.
Yes, and I want to add to that as well that I know many of our families know, but for anybody in our audience who may not be familiar with this, that we do have, and we see families that have children, siblings that are all affected with a peroxisomal disorder. They all would have the same identical mutations, and they each clinically present very different.
Right.
And
right.
So there's definitely the unpredictability. As a parent myself, it's very frustrating. It's one of the things that we wish we could be able to have more information on. But even amongst siblings who are affected, we definitely see them present differently.
Right.
And I'm not sure I'm going to say this correctly. Have you seen any correlation with Heimler's and is it Shari malformation?
Okay. No, I haven't. That's interesting. So I mean, the answer is no. I have not seen it. I don't know. I guess if I was betting, I'm not sure I would relate them, but I don't know. Heimler is an interesting kind of counterpoint to the terminology discussion. There is some discussion amongst us. I think most of us would like to term the individuals who receive, the diagnosis of Heimler as mild PBD-ZSD. But it is interesting. Some of the individuals with Heimler seem to sort of have isolated, dental and nail issues, and those are due to the same genes. But Chiari is kind of a brain, malformation of the kind of hindbrain isn't known, as far as I know, to be connected.
And I know you briefly mentioned this in your presentation actually last year at one of our conferences about the misdiagnosis sometimes of other diseases. And I know that with the new way of diagnosis, the way things are happening, we're starting to see that a little bit more often. So Heimler, I'm seeing that word a lot being used as a diagnosis rather than the overall spectrum terminology when we have someone who may have an Usher diagnosis that was not genetically diagnosed.
Right. We had an interesting patient that was diagnosed with Usher, and we ended up finding PEX1 G843D homozygosity on a sequencing studies that we did, and that was sort of an interesting variation on how people get diagnosed. But I think the more we can, I guess I'm more, in favor of grouping individuals based on the genetics, and then, but then just acknowledging the wide spectrum. And
I'm looking to see if any other questions, please get them in if you have any other questions. I'm going to ask one more, and that's because I've seen it, and I'm a family who has a son who was diagnosed back in 2001. And at that time, we were not able to identify the mutations, or as we now say, variants. So I'm curious, I know we spent a lot of time, and I really appreciate, and I'm sure everybody else does too, that you didn't take it easy on us. And you spent a lot of time explaining this, because we need to hear this. And I know for me, we weren't able to find the variants for years. My son was diagnosed biochemically. It was very in the old school way of having a lot of testing done, trying to figure out what his diagnosis was, and going through not one, but two fibroblasts. And so we had a pretty long diagnostic odyssey. And so for those of us who are a little old school, we know that we didn't have to necessarily see those variants identified to receive a diagnosis. What would you recommend to a family today who may have a child who they're looking at this clinically and saying, this has got to be a peroxisomal disorder? And then biochemically, there's the elevated very long chain fatty acids, and they're thinking, yes, this is a peroxisomal disorder, but they can only identify one pathogenic variant. What's the next step for that family? Do they have a diagnosis?
Okay, great question. And actually, I'm going to split it into two parts, because one thing to point out is that I've had this experience with other disorders as well, where maybe an individual is diagnosed and we don't even know the gene at the time that they were diagnosed. And then, we come in with a new diagnostic criteria that calls for the gene diagnosis. And, I had a specific case where the family was, it was sort of like, this is what we've known our whole life, and you're telling us we don't meet the criteria anymore. So I think when you're applying modern criteria to sort of an individual that a lot about, we know a lot about an individual who's in their 20s, 30s. We can know a lot about their features. So we can sort of look at them clinically, and we can look at their story, get their records, and we can, tell the difference between a mild Zellweger spectrum and, another disorder. So I think that's a different scenario than a newly diagnosed infant, for instance, where you really want that, you want the ability to predict. So if you want the ability to predict, I would love to get the mutations. But yes, you're right. Then even in that group where I want to get the mutations because I'd love to be able to see if we've seen these mutations before, I still would say that the mutations are not required for the diagnosis. If the patient has, consistent features, and if we have elevations in sort of the biochemical studies, there's other things we can do to, confirm. Maybe we would do fibroblasts in a few of those cases, but you don't need the mutations for the diagnosis per se. It's more just that if you're in a situation with a newly diagnosed individual, it's great to sort of have them to help predict. So I think that's kind of the issue.
Awesome. Dr. Wangler, this is Melissa Bryce Gamble. I was just going to ask a question about your Metabolomics project as we wrap this webinar up. We've had a lot of conversation tonight about variants and, the correlation between genotype and phenotype. I'm wondering if you believe that the Metabolomics project that you've been working on, that the GFPD funded, could help identify some modifiers or some different things that may help explain some of that variation that we see of siblings even that have the same variants. See, we're all struggling to get from genetic mutations to variants. So yes.
Yeah, so it's a great question, Melissa. So what happened in that project was, so we had about, I think 19 individuals, and we've added more, of course, as time has gone on. And then we added the RCDP cohort that you guys teamed up with Rhizo Kids on, along with the Wynn Matafee Foundation. But what happened in that first round was we wanted to answer the question you asked because it would be fantastic. Like if we could sort of use metabolomics to give us some insight into severity that even might act as an intermediate between the gene changes and what happens to the patient. But what we saw in the data was that age sort of overwhelmed all the age, explained so much of the variability that we needed. What we need is sort of just more more samples. And then that's sort of the subject of the grant that we wrote this past year to the NIH that we think we may have good news on, as long as COVID doesn't change everything about the federal budget. But that would allow us to maybe get bigger numbers and start looking at your question. So I guess the answer is we weren't able to answer it because age overwhelmed the variability.
Right. But that just speaks to the point that we want our families to be involved in research, and we hope that there will be a way that we can explain some of this in the future.
Right.
Yes.
And answer those questions that our families often have about severity and progression. We just don't know yet.
Right. I mean, and I think it's sort of like you're going to have to do a lot of, I think it's metabolomics could be like part of it, but there's, probably, it's also the clinical part, the natural history part, in terms of sort of what Nancy's doing, in terms of defining how you're comparing the cases. And so.
Thank you so much for taking the time to answer all of these questions for us.